It's my absolute pleasure to introduce the next awardee, Val Smith of the University of Kansas, for the Martin Award on a paper with the very high impact on aquatic sciences. Val's one of the most humble, honest, yet forthright scientists I've ever met. We've all got our scientific heroes, and Val's undoubtedly one of mine. We heard in the plenaries recently, the climate plenaries, that there's often interesting phrases that are used to describe people. Flatulent demagogue was the one that jumped to mind. I'm going to go with cherubic genius with Val. He's got a picture in a closet somewhere that's aging badly. Val is a terrific scientist who's absolutely generous with his time and his insights, and he's critical yet always kind, and I think he's a model for all of us. At any rate, he's going to talk about his paper, but I just want to say a couple of brief words. In his nomination letter, David Schindler's pointed out that the paper's been cited nearly 800 times, making it one of the most highly cited references in water quality degradation or eutrophication, and it's really been one of those instrumental papers that when you look at it now, you kind of go, yeah, we know this. But at the time, in context, this paper was absolutely transformative, making us focus on the primacy of phosphorus pollution as a causal agent in the degradation, but also making us think about things a little more in a more complex fashion, looking at the stoichiometry, the relationship between elements, and how that affects the community composition and how the public perceives water quality issues. So with that, I'm going to introduce Val Smith, and please join me in welcoming him to the stage.
Well, it's impossible to overstate the impact of this award on me. I'm just completely humbled and amazed that it's happening. I owe a lot of thanks to an awful lot of people, a number of whom are in the audience. I certainly appreciate the committee, and of course I've been a member of ASLO for a good long time, and it's just an immense honor to be here and to receive this award, and I thank you. With that, I'd like to have the first slide, please. This didn't all occur in a vacuum, and like Curtis Settle, I'd really very much like to kind of indicate the origins of these things. I was a bit more hirsute in 1975 when I joined Joe Shapiro's lab at the University of Minnesota. And I was there with a group of truly outstanding colleagues, one of whom, right there, Mike Lynch is one of the world's finest evolutionary ecologists now at Indiana University. This is an incredibly rich intellectual environment, as all of you know. Joe Shapiro was a student of Hutchinson, G. Evelyn Hutchinson, and so he regaled us with stories of Hutchinson all of the time that we were there, and it really set a milestone or set a yardstick that we always tried to emulate. We always wanted to try to do our very best in being part of the lineage of G. Evelyn Hutchinson. 1 year after I arrived at Minnesota, another very important personality arrived as a new assistant professor. That was David Tillman, who is a very good friend and an amazing colleague and became my co-advisor. Dave, as you know, is the originator of resource ratio theory, and which now underpins much of modern ecological community and ecosystem ecology. And it's hard to imagine a finer group of mentors than these two. Joe Shapiro was Canadian, and so I felt Canadian roots from the very beginning. And soon after the publication of my first paper, I came on the radar screen of Frank Rigler, who had moved from Toronto to McGill University. It turns out Joe was a McGill grad. And very soon, as I was finishing up my PhD work, Joe began making moves to try to get me a good position elsewhere. And I landed a NATO postdoc with Frank Rigler at McGill. Tragically, just a few months before I actually physically arrived at McGill, Frank passed away unexpectedly to terminal cancer. But I inherited his research project up in the subarctic of Canada, and I completed that. But before I actually arrived, years before I arrived at McGill, I had kind of an inkling of what was going on at McGill, because one time just before Christmas, I was sitting in my office in Joe's lab, and I felt a tap on my shoulder, and this voice says, "Hi, I'm John Downing." And I said, "Oh, hi, John. Nice to meet you." And it came to pass that, of course, John is from St. Paul, and he trundled over. I was apparently on Frank's radar screen, which meant I was on the McGill Group's radar screen, and he came to say hello, and we've been steadfast friends since that time. Great friends. Much appreciated. And he gave me a lot of these photos, by the way. So Frank left behind a great group and a great group of mentors. This is Jaap Kalf and Rob Peters, after experiencing one of those days from hell in the field. But they had an amazing group that I joined as a postdoc. And I think that you'll see here some people who are well known to us. Robin, one president of ASLO, a very young Carlos Duarte with his daughter and wife. Possible next president of SIL. Alain Vézina here is the director of Bedford Institute. This was an amazing group. These people have gone on to do wonderful things. This is also a wonderfully important intellectual ferment in which the paper for which I'm being honored came to pass. But I shouldn't fail to mention that paper was vetted by, at the time, one of the most keen intellects I felt in aquatic science on the planet. And that was Richard Vollenweider, who was at the time at CCIW in Burlington. And I grew to know and like Richard, and we spoke by phone for decades after I left Montreal up until the time of his death. Truly a wonderful man, and his monographs and really set the stage for understanding and modeling and also management of global eutrophication. But I named two people, Joe Shapiro and David Tillman, who were important in my formative career. But then there's also this guy. So this is a very buff David Schindler from, I think, in the LA in 1981 or so. And Dave and I met, I believe, in the early 70s. He came to give a seminar at Minnesota, and we corresponded for years. And I still have handwritten data sheets. When I was forming some of my early ideas and needed data, Dave sent me sheets of data. He said, "Here, take these. Test your hypotheses." And so if you like my paper, thank Dave. If you don't like my paper, well, this is all his fault, right? So we share an abiding love in these kind of cyanobacteria, these things that really crud up our lakes and reservoirs and water bodies all across the world. And Peter was correct. At the time that I was thinking about these, our understanding of community ecology was minimal. Our understanding of the factors that regulated not just the abundance, but certainly the species composition of phytoplankton communities was trivial. And Joe Shapiro had only a few years before, and my paper came out, had said that he felt that CO2 was the key. Now, Dave had an interesting paper, and it's a little bleached out, I apologize for that, in 1977, published in Science, and this kind of made Joe cranky because he thought that CO2 was the answer, and he didn't really believe this much at all. But I, as a young person who was still forming his ideas about things, took heart from this and thought, this is very interesting because all Dave had done was alter the NP ratio in the fertilizer in this lake, and Lake 227, and he found all of a sudden you had this appearance of infixing cyanobacteria where they had not been before. So I got interested with NP ratios. Dave Tillman's there with the NP ratio, resource ratio theory, and I decided to test some of these ideas empirically. So I set out for several years to collect the very few data that then existed all over the world that I could actually access. And this is what I created. And this is basically at the heart of the paper that you know about. Several key features. First, of course, is this apparent threshold at about 29 to 1 by mass of TNTP, at which it appears as if there's a break point. So below that, you tend to have a high abundance or high dominance by cyanobacteria, above that negative. Okay? But I want to point out that on the left side of that line, you have almost equal probability of having 0 or 100. So there's some sort of a switch, and this noise is important. Okay? Similarly, over here, it also is not a fixed threshold because there can be cyanobacterial blooms at ratios above this. And actually, as I began to collect more and more data, I found more and more examples to the right of this line that made me think that, well, okay, it's a little bit more fuzzy than this. Okay? It's not a make or break. It's not a critically sharp threshold. So I began to analyze more and more data, collect more and more data, and this paper is less often cited, but in this paper I looked at the interaction between nutrients and light and found that, well, yes, NP ratio did have an effect. Okay, that's here, and this is percent blue-greens, but this is basically in a model. The brighter it gets, the less likely you're going to have blue-greens at all, regardless of ratio. But as you eutrophy things and light climate gets very dim, then you also are going to have more blue-greens. So absolute quantity of nutrients matters because it affects light and transparency, which also interacts with NDP to dictate which kinds of blue-greens you get. Okay, so you can have blue-greens up in here, even at high ratios that far exceed that 29 to 1 number. Okay. Some years later, there have been hundreds of papers talking about NDP ratios, resources, CO2, and various reasons that are been put forth to account for why there are blue-green blooms and blue-green dominance. Jim Meltzer has a very, very nice paper, and I like Jim's language here. Viewed in this way, cyanobacterial blooms can be seen as probabilistic events that are the end result of a series of key mechanisms involving nutrient loading, physical mixing, and trophic interactions. So looked at it in that way, he had this kind of a decision tree. And the thing to take home from this is, I'm not wedded to NDP. What I did was attempt early on to try to point the way towards NDP as being a primary driver of cyanobacterial blooms, but I'm not wedded to be, and I certainly never say it's the only factor. It's actually multivariate, multifactorial. Okay? John, Susan Watson, and Ed McCauley had a paper in 2001 in which he was really emphasized the role of the magnitude of nutrient loading as opposed to ratios. So, and John very nicely said, we should be testing alternative explanations for the dominance of cyanobacteria. You saw that left-hand side there. There's huge variance, so we need to understand all those factors, okay? Just last year, we had a very nice study. Arguably, I think this is a superb study, looking at the NP ratio implications of cyanobacterial toxins. So microcystins are very nasty toxins associated with cyanobacteria. And here they actually looked at the effects of NP ratios in the water column on, now this is concentration of microcystins in micrograms per liter. So here we once again have this kind of a threshold look. And even if you take it way down here, there's still this hint that yes, NP does matter. And this is from all of these 9 different provinces all across Canada, eh? So I would argue once again that there seems to be a strong suggestion that N2P matters. And in fact, if you take these data and you calculate the probability of exceeding certain concentrations, so for example, 1 microgram per liter is the WHO drinking water standard, then as you increment the NP ratio, that risk goes away. So once again, we see this signal coming through. But I'm sure it's not as simple as this. Shortly on the heels of my paper, or that paper, this paper by Dolman et al. Suggested that cyanobacteria should not be treated as a single group when considering the effects of changes in loading. Going back to my original study, I lumped N-fixers, we didn't know much about cyanobacteria at the time, and non-fixers. I still got the pattern. But this is a point well taken. We really need to know much more than we do know about the behavior of N-fixers and non-fixers. Now, this also leads, it was mentioned both by Dave in his comments about my award and also by Peter. I like to think that this paper helped very much in establishing phosphorus and phosphorus control as a cornerstone that we must continue to use in eutrophication management. And Dave has hammered this again and again. I firmly believe that absolutely that a cornerstone of modern eutrophication control, freshwaters especially, is phosphorus control, right? Having said that, I also note there's increasing evidence that I already indicated that this is a multivariate phenomenon. All of these effects of P take place in a larger context, in many cases of changing N. So over the past 40 years or so, one of the things that's really been happening is we've been also, anthropogenic N loading has really greatly increased. There's huge amounts of N. So we've got a different condition than when nutrients were primarily brought in by point sources. Now we have non-point sources. So we've got a different context. And it looks increasingly like that we need to look at both of these elements. And I'm hopeful that as time goes on, the scientific method will show us the way. And in closing, I'm trying to keep this short because I know we've got a break coming up. I do want to once again thank everybody. I'm indebted to Dave for almost a lifelong influence on my science. And I'm truly humbled by this award, but I'd like to close it this way. Thank you.
Anybody have any quick questions that they'd like to make to Val or comments?
I have a comment, and that is that I think a lot of the confusion over NNP has been over this point that Val made, that a lot of those early papers just lumped the nitrogen fixers, which have an advantage in low nitrogen situations, and the non-fixers that don't necessarily. But when you look at the whole ecosystem scale, I still have never seen a lake or an estuary where nitrogen has been controlled where it made any damn difference. And if anybody has such a case, please send me the paper.
Yep. I think it's only through data and empirical evidence that we actually will tease this out. And so I'm absolutely on board with Dave. We really need more information on how these systems respond to P control only, N control only, and both. And then we can begin to tease out exactly how best to manage these ecosystems. Lissa?
Val, you served nobly on the Academy's Algae Biofuels Sustainability Committee. For those of us who are going to miss your talk on Friday, would you give us a little sneak preview?
Oh, yes. Lissa is, I believe, the CEO of Live Fuels and very much interested in algal biofuels. And as she indicated, I was honored to be a member of a 15-member panel to look at the sustainability of algal biofuels. And if people who do wish to come on Friday to my talk, one of the things I've always done applied and basic research. And one of the things that I've been really happy to do in the last 3 years or so is to take some of my energies and move them into trying to transition the world into alternative liquid biofuels that are created from algae, because they have very rapid growth rates. But one of the problems that I've found is that I believe that many of the systems that are currently being formed right now to create these algal biofuels and the consortia that are put together, some of my members on the Academy panel really do believe that there's too little ecology and there's too little biology. And what I intend to do on Friday is to invite aquatic scientists who are knowledgeable in population, community, and ecosystem ecology to join and partner with and inform our efforts in algal biofuels. Thank you, Lissa.
These folks should know that there were, the word in our industry was there were some knockdown, drag-out fights on that committee. And Val, thank you, as I said, for serving nobly.
Thank you. Good to see you.
How much do
Hi,
we
Frida.
know? Hi, thank you. How much do we know about the transfer of nitrogen between the nitrogen fixers and those blue-greens that are not nitrogen fixers but benefit somehow from that fixation? What are the mechanisms of transfer?
Well, you're talking to the wrong person, I'm afraid, Frida. I'm not up on that literature, but certainly DON, which would be presumably leaked by some of these fixers, is going to be there for uptake and processing by many portions of the microbial communities, and that could transfer the DON into bioavailable inorganic N, but we really need somebody who's more knowledgeable than I am to answer that question. It looks a little bit like Dave again.
Yeah, that question, I think, is a really important one, but it's not only a transfer of the N to other cyanobacteria. One thing that some of the greatest names in limnology have missed is that once it's fixed, that nitrogen sinks, and over time, it increases in that system. It doesn't have to, as they say, fulfill the entire nitrogen requirement in a year. It takes 15 or 16 years or more to build up to where it is, but it benefits all the algae.
Okay, cool. Well, I'm getting the cutoff note because we got other things to do, so many thanks again to the Society and everybody for coming. Take care. Thank you.